actual process, the question of how do we get from the DNA to the RNA? We used to think that, and I've mentioned this before, we used to think that the vast majority of our DNA was junk DNA. People used to actually refer to it as junk DNA. But we've since learned that about 80% of the genome is functional, doing really important functional things. And we've talked about a lot of those things. We've talked about that there are a lot of RNAs that are doing really, really important things to keep our bodies functional. And when we talk about inactivation in a bit, you'll see that, how important an RNA is that is not transcribed into a protein. Just reminding you of this once again, we're going to be focusing on proteins, but there's a whole lot of the DNA, a whole lot of the genome that's doing important things that is not necessarily going to be translated into a protein. Okay, also, I focus a lot on eukaryotes, and we're mostly going to be talking about the eukaryotic genome. But I want to orient you to a prokaryotic genome so that you can see those contrasts. So over on the left of your screen is a prokaryotic cell. So a prokaryote, if you remember, does not have a nucleus. So this sort of blue object, this sort of blue wavy thing here, that would be this prokaryotes, what we call circular chromosome. You can see it's continuous rather than where ours are discontinuous. Ours are sort of like lines. There's an end and a beginning. Prokaryotic DNA is circular. And what this image is showing you is that the DNA is being transcribed as the same time, if you look. So that the purple is transcription, and then these ribosomes, what they're doing is they're translating that mRNA. So this would be translation. So transcription and translation in prokaryotes, because they don't have a nucleus, can happen simultaneously. It can happen at the same time. So as RNA polymerase is producing the mRNA, ribosomes are translating it, which is really pretty stinking cool, I think. As opposed to eukaryotes, we are eukaryotes. You are a eukaryote, if that helps you remember that, where we have a nucleus. And this cell is showing the nucleus super, super enlarged so that we can see what's going on. This is just another case of, depending on what we're focusing on, our visualizations might be a little bit different. And it's showing you, so these lines are what's called the nuclear membrane or the nuclear envelope. Just like a cell has a plasma membrane, so does a nucleus. And then these little openings, these are nuclear pores. That white doesn't show up very well. So these openings, these are nuclear pores. And in this image, it's showing them very, very open, but they're incredibly, incredibly restricted. So what can and cannot exit and what can and cannot enter is tightly, tightly regulated, as you might expect. So what this is showing then is that the DNA, pardon me, the DNA here is inside the nucleus, as I've emphasized, and then the RNA polymerase, so we could draw like a little RNA polymerase on here, is producing what we've learned is the pre-mRNA. That's this right here. It's producing that pre-mRNA. And then that has to be processed to become mature mRNA here. And only the mature mRNA can exit the nucleus to be translated. And it's translated out here in the cytoplasm. Just so that you are aware of where we are, and I will emphasize this again to make sure that you are aware. Okay, so some sort of, I guess we call them basic rules of conduct with the genetic code, and you're not using this book, so ignore that. And I'm going to step through these super fast because we are going to talk about them individually, slowly, one at a time. The genetic code is written in linear form. Each ribonucleotide in the mRNA is equivalent to a letter, a complementary letter in the DNA template. Each word in the mRNA consists of 3 letters. These are codons. We're going to talk about this more when we get to translation. Each codon specifies either an amino acid or a start or stop, telling the ribosome. So codons, we should tell that, codons talk to ribosomes / tRNAs. So they matter during translation. It's easy to get mixed up about when does what matter. So codons are important in translation. And it's unambiguous. So any one codon can only specify one amino acid. So one word corresponds exactly to one amino acid. But it's degenerate because a given amino acid can be specified by more than one codon. If you're like, what? That's so confusing. Don't worry. When we get to translation, we are going to dig into that. Collinear, so there's no internal punctuation. Each codon is translated in a linear order from start to stop. And they're non-overlapping, and it's nearly universal. Almost this same code is used by almost all life on Earth. Once again, emphasizing, we started with the DNA. It's packaged as chromatin inside the nucleus, euchromatin or heterochromatin. If it is getting transcribed, so this is going through transcription. If it's getting transcribed, what kind of chromatin is it? Okay, thinking back, euchromatin, heterochromatin. Remember that heterochromatin is the chromatin that's super, super tightly packed, and this is for non-active DNA. DNA that's being actively transcribed is going to be packaged as euchromatin. Transcription, we know we talked about this already, translation into at the ribosomes. Transcribed into mRNA, and then the thing that I'm emphasizing here is that all of this happens inside the nucleus, and then all of this happens outside of the nucleus in eukaryotes. I'm going to take that off so that you can see that. Okay, we're going to be focusing on here, stuff that happens inside of the nucleus. We're interested in transcription right now. Once again, DNA is the instructions or the blueprint, and RNA is the messenger. So some transcription terminology. The promoter is a regulatory region immediately 3', which means upstream, and there's going to be a whole video that goes through this. So if you're not catching it right away, that's okay. So 3' is upstream of the transcription start site of a gene. So this whole area is the promoter. So it's upstream of the transcription start site of the gene. The transcribed region is this whole area. So the transcribed region then is downstream of the transcription start site. So if transcription starts right here, the transcribed region is downstream, whereas the promoter is upstream. Okay, within the promoter, there's an area, it's indicated here. There's an area called the consensus sequence. So this is embedded within the promoter, and it's a highly, highly conserved sequence that helps RNA polymerase bind. So this is where RNA polymerase is going to bind within the promoter, is at the consensus sequence. In bacteria, just a little information, bacteria have 2 different consensus sequences, and these are your This is just some information about that and kind of where they are relative to the transcription start site. Just notice that there's variation. So this top consensus sequence is only 10 base pairs from where transcription actually starts, whereas this bottom one is 35 base pairs from where transcription actually starts. So that's just showing you that there can be variation in that. Consensus sequences are an example of what's called cis acting elements. We defined those briefly earlier, and I'm on the next slide. I'm going to show you a little bit more about that. So these are sequences that are close to the gene whose expression they influence, and then transacting factors. These are a different thing. Transacting factors are usually products of genes elsewhere in the genome. So transacting factors are usually far away from the gene that they act on. So I'm just going to give you a visual for that because those are going to be really, really important when we start talking about transcriptional control. So if this is our transcribed region, and this is our promoter, embedded within the promoter will be a consensus sequence. It's going to be a consensus sequence. This consensus sequence right here, you can see that it's physically close to the gene that it influences. It's actually part of the gene that it influences because this entire thing is a gene, not just the transcribed region. Okay, so this is a cis-acting element. And then a transacting factor is going to be something that comes from far away, and interacts with this gene in some way. So that's a transacting factor. And these can be RNAs or they can be proteins. One example that we'll talk about a lot are called transcription factors. And these are proteins. Okay, and then the last thing, RNA polymerase. RNA polymerase is the enzyme that synthesizes the mRNA transcript. In eukaryotes, there are 3 different types. We'll just talk about RNA polymerase in a general sense, but you might, if you're using your resources, if you're seeking out videos and information, which is great to do, you might see them talking about RNA pol 2. And this is the RNA polymerase that does the work of transcribing mRNA. Other RNA polymerases transcribe other types of RNA.